Okay, we left off with the large intestine. We said, if you remember, I said there's 6 parts to the large intestine. We talked about one right here, the cecum, because it's the beginning. Off of the cecum is the appendix. And we said where the small intestine meets the large intestine, there's a junction called the ileocecal junction. And it gets its name from the cecum and the ileum, the last part of the small intestine. There's a sphincter there called the ileocecal valve, not a sphincter, valve. Then we go up. Remember, this duodenum goes to this part right here. We go up, and that is called the ascending colon, sometimes called the bowel. Then as it crosses right underneath the diaphragm, it's called the transverse colon. Here's where I'm at. Ascending, transverse. Then when it bends down, it's descending. Descending. And then, now I drew it as a straight piece here, but actually what it does, it makes a little S turn, like this. And this part is called the sigmoid colon. And then it swells and has a sphincter on the end. Sphincter, as we know, is the anus. You know a few people like that. This is called the rectum. And that's for storage of feces. So sigmoid, S, rectum, anus is where the digestive system terminates. Okay? Now, I'm going to take you down here. We're going to draw some other stuff. Remember when the small intestine, and I was talking about segmentation and peristalsis? In the large intestine, it's more dramatic. You're going to find out these are called haustra. They're more dramatic than they are in the small intestine. Small intestine, they're very small, so they kind of look at it as smooth. The same thing happens with smooth muscle, is it propels food along this way, called peristalsis. And it also churns it. And that churning is called segmentation. We already talked about that. Each one of these sections is called a haustra. Each one of those sections is called a haustra. The rise in the haustra is called diverticula or diverticuli. So we're talking about a section of the large intestine. We said a section is called a haustra. The cecum was the first part of the large intestine. We said a rise in that haustra is called a diverticula or diverticuli. And there are times when, because of pressure placed on the rectum or the sigmoid colon, that this will pop out like a little balloon or a pouch on the outside. When you have that, that is called diverticulosis. When the diverticulosis, which is that pouch, okay? Remember, diverticula means it's just the way it's supposed to be. When the pouch is there, it's a condition called diverticulosis. When that becomes inflamed and possibly bursts, that is called diverticulitis. These are the terms you see right here. Diverticula or eye, diverticulosis, pouch, diverticulitis, inflamed. Good. We talked about the 6 parts: the cecum, ascending, transverse, descending, sigmoid, rectum. Anus is just a sphincter at the end. Now, picture a garden hose. You got me? I'll do it over here. Picture a garden hose.
Give me 1 second, I'll zoom in for you.
With no water. You got it? With no water going through it. Now, you know, if I turn a blast of water on, that's going to straighten out. The same thing with your intestine. You've got these ripples called haustra and diverticula. If pressure comes around there, they'll straighten out. So what we have to do is we have to put smooth muscle along a ridge to hold it in place. And the smooth muscle is called tenicoli. Tenicoli. Here's what it looks like. You see this white band right here? That white band represents smooth muscle called tenicoli, and it holds the haustra in place. Tenicoli. Somehow I'm going to get that on your lab exam. Now, another thing I want you to think about is we have 2 types of muscular systems in our body. One is the ones we have control over, which we know is skeletal muscle, and the other one which is autonomic, which is smooth muscle and cardiac. If you go to sleep, your conscious part of your brain puts your body at rest. It seems like you would defecate yourself, which means you have to have smooth muscle involved because it's going to keep constricted. But we also have to have command of our anus to tell it it's time to defecate, which means it has to be skeletal. So that means there's 2 muscle types down there. The answer is yes, there is. The internal anal sphincter is made out of smooth muscle. It's the one that holds things in when we're sleeping. The external anal sphincter is made out of skeletal muscle. And they communicate back and forth when we're defecating. Okay. That's it. No. That's it. We've connected. We started with the mouth and we ended up with the anus. Okay. Remember then I said there are glands or organs that help us digest food by secreting digestive enzymes. In your book, it'll say teeth, pancreas, liver. We're not going to say teeth because that's in that mouth to anus. We're going to do pancreas and liver, and we're going to start with the liver. It's called accessory glands or accessory organs. The first one is the liver. This is the liver. You can see there's a white band. This represents a ligament. The liver is very heavy. Sitting right where my hand is your diaphragm. Okay, so that's the upper wall of the abdominal cavity. And it separates the abdominal cavity from the thoracic cavity, which is right here. Since the liver's heavy, it has to be held up by a ligament. This ligament right here is called a falciform ligament. Falciform ligament. And that's not enough. You can see there's a round, we asked this on the last exam. There's another one right here, which is called the round ligament. And that holds the liver to the front of the abdominal wall. Round ligament, falciform ligament. Holds the liver in place. That's why you see falciform ligament, round ligament here. Okay, still dealing with the liver. Hepatic means what? Hepatic means what? We've talked about this. It means liver, portal, a one-way tube, a one-way tube. Remember we talked about it when we did the lab on cardiovascular, and it was question number 5D as in David. I said, I want you to read about it and explain. It begins and ends in what? And the answer should have been capillaries. And it takes digested food, remember the pearls we've been talking about, from the small intestine to the liver. And what happens, they bring those digested foods to the liver, it's cleansed or separated, and then it's dumped in the blood on the other side. So a hepatic portal system begins and ends in capillaries and is for transport of digested food to the liver. Okay, now, down here, I'm going to draw the liver. This is your liver if it was in me facing you. Falciform ligament, round ligament. Falciform ligament, round ligament. Right here is the gallbladder. For storage of bile. Now what I'm going to do is I'm going to lift that liver up like this so you can see the bottom of it. We lift it up from the bottom, it looks something like this. So this here and this here are the same. Your liver is lobed. Let me show you on the model what I've just drawn for you. See it right here? So I drew it once like this, and then I said if you look at it from the bottom, it would look like this. And that's what I've drawn. Your liver is lobed. Your liver is lobed. And each one of these lobes has a duct that comes in to the gallbladder duct. And it's not called the gallbladder duct, it's called the cystic duct, but I haven't got there yet. So I'm drawing the different tubes that lead to that duct. I'm going to change colors for a reason. I need one more color. I got it. We'll make it black. Now there's three colors here: green, orange, black. Blue, I guess there'd be four colors. Blue is the hepatic ducts. Blue hepatic duct, not hepatic portal, hepatic ducts. One of the functions of the liver is to break down red blood cells. We talked about this when we did blood. Remember? And we said embedded in the phospholipid bilayer of the red blood cell is an enzyme called hemoglobin. And because the most common cell in the body is red blood cells, and they have a life expectancy of about 120 days, we're constantly building them up and breaking them down. This is the guy that does most of the breaking down, the liver. So it's going to take red blood cells and hemoglobin and break it down. When it does, it's going to use the breakdown of that, which is called bile, to emulsify fats down here in the duodenum, which is why I left that up there. This gallbladder, this duct that I drew in orange is unique. It's called cystic duct. Black one is the one that goes all the way down to the duodenum, and this is called the common bile duct. Common bile duct. Now, the orange, the reason I drew the orange there, and probably should only have drawn it right to here, is the cystic duct is a two-way duct. And I'll do it in red. Red broken down, red blood cells are being broken down, is taken to the cystic duct where it's stored in the gallbladder. It also, when it releases it, it sends it down the cystic duct to the common bile duct. The common bile duct is a one-way duct going to the duodenum. Hepatic ducts are a one-way duct going to the cystic duct. Do you see? So on your exam, I'm going to talk about that. Cystic duct is a two-way duct leading into and out of the gallbladder. So let's go do a little checking off over here. Hepatic ducts, gallbladder, cystic duct. We good with that? And we said bile, this is a test question. Bile is for the emulsification of fats. As you remember, fats are hydrophobic. So when bile is added to them, they become hydrophilic in a way. So water can mix with it, and enzymes can mix with it. Okay, we said red blood cell breakdown, most of it occurs in the liver, and we break it down into iron, the heme, globin, the protein, and that gets us bilirubin. And I'm going to talk about that after we get through this and take a break. Now, right in the lesser curvature is the second accessory gland, kind of looks like a bird's feather. And that's called the pancreas. It's called the pancreas. Here's our map. Pancreas. I'm sorry, I said lesser curvature. I wanted the greater curvature. Greater curvature. Lesser curvature is here. Okay. On the other model. Here. The white you see is the pancreatic duct. Now, I'm going to come back to CCK. The pancreas, which is our second accessory gland, has two functions. This is on your exam. It is endocrine and exocrine. Endocrine means it produces hormones. One common hormone that you're familiar with is insulin. And it's also exocrine, produces digestive enzymes. So the pancreas, two functions, endocrine, exocrine. Now, remember, I'm going to take you back to when we were talking about the chyme. Remember we said when it comes out of the stomach, it comes out through a sphincter called the pyloric sphincter. And we said the pH is right about 1 to 2 because, as you remember, we said that you have to break down proteins into amino acids. And one of the most difficult bonds to break is a peptide bonds of proteins. So when it comes out of the duodenum, it comes into the duodenum, pH is 1 to 2, which means anything in its way is going to eat up with acid. So we have to neutralize it. So the pancreas, as you can see right here, the duct of the pancreas is in the very top part of the duodenum, just like the common bile duct. They come in at the same point. But that means food that comes right out of the stomach into the duodenum is hit with bicarbonate, sodium bicarbonate, which brings the pH back up close to 7, which is neutral. So what happened is you have coming down here, and I'll do it in green, you have a pH 1 to 2. Right here, and we did it in red, so it'll be consistent. Right here is a duct, and that duct dumps in. We said it has endocrine and exocrine function, and one additional thing is to bring in something to neutralize that pH, which is sodium bicarbonate. Hope that made sense. Come out of the pyloric sphincter, your pH is way acidic. You have to neutralize it or it'll destroy the cells there. So you neutralize it with sodium bicarbonate, which is released from the pancreas. We're also going to see sodium bicarbonate released from the cells of the duodenum too. Now, remember, we've talked about this before. We said when something is really important for your survival, we put a sac around it. So since your internal organs are important for your survival, not internal organ, your abdominal contents is important for your survival, we're going to put a sac around it. And that sac is called the peritoneum. And it has 2 layers, just like every sac we had, except for the one around the brain. We'll see the next third. The outside is called the parietal. The inside is called the visceral. Yes? And we know the space in between bears the same name as the sac. So the space in between is called the peritoneal space. Okay? It's full of a fluid. Peritoneal fluid. I didn't put that up there. Peritoneal fluid. Now, not all of these glands or organs are contained in that sac. There's a few of them that are behind it. And we know the Latin word for behind is retro. So retroperitoneal means the glands or organs that are not in the peritoneum. And that's called retroperitoneal. Example, kidneys. Example, kidneys. Now, to do a little more cleanup, the peritoneum gives rise to some extensions. I think we've all seen in a shot of a military trucks and jeeps and stuff like that, they put a netting over it. And that netting has woven camouflage, almost like pieces of leather or rubber or material. So up above, when they look down, they can't see it. Okay? You have a netting like that inside your skin on the outside of the peritoneum, and that's called omentum. Omentum. And you have a greater and lesser omentum, but omentum is what I'm after. It's extension of the peritoneum, and it's for storage of fat. Storage of fat. Another extension of the peritoneum is mesentery. Now, if I took all the intestines about 25 or more feet, and I took it, I cut somebody open, and I put it into a bucket and did some stuff, whatever I had to do inside, I could pick it up, drop it, and it would go right back into that shape, which therefore means it has to be held in that shape. So it kind of looks like my hand sticking out, intestine attached to it like this. This part that it attaches to, called mesentery. Called mesentery. Now, what I usually do when we have in-class time is when we do the cat dissection at the end of the semester, we cut the esophagus we cut all of the intestines out, all the way down to the anus, and we stand and we pull it. And you can feel it pull away from the mesentery. And then we can look and see where the mesentery had attached to the intestine to hold it in place. So now, two extensions. We talked about two extensions. One, mesentery. Two, omentum. Another term, cirrhosis. You've heard of cirrhosis of the liver. Immediately you think of drinking, alcohol. Could also be drugs. It could also be subjected to poisons. And what happens is your liver, to make it simplistic, your liver is to filter toxins out of the blood. When that happens, the cells of the liver, which are called hepatocytes, they could be killed, I guess is a loose term for it. They could be sacrificed. The internal cytoplasm of a hepatocyte becomes so toxic that the cell cannot carry out its normal activities and dies, which means we have to go through mitosis to replace it. If this is happening at such a fast rate, then we start losing cells, and they cannot undergo mitosis, and they build up a protein wall. In other words, where there was once a nice healthy cell doing what it's supposed to be doing, it forms a protein scar. When your liver is scarred, it's called cirrhosis. So cirrhosis is the scarring of the liver. In other words, it just has proteins there that don't do anything but hold cells together. So a cirrhotic liver is a scarred liver, and we can get it for different ways. Hepatitis is a viral infection. All right. Is a viral infection. Now, when a virus enters an organism, it cannot reproduce on its own. It has to use the organism's cells to reproduce the virus. So what it does is once it gets inside your cell, it goes to your DNA, takes over the DNA, and tells you you're no longer making a liver cell, you're going to make me. The viruses have only certain cells that they can attach to. As we know that with this coronavirus, the virus attaches to lung cells, not simple squamous, but cells that produce, let me think of what it's called. Angiotensin converting enzyme, ACE. It's a cell in your alveoli that produce an enzyme that converts angiotensin 1 into angiotensin 2, which I'll teach you about when we do the excretory system, or you take physiology from me. Regardless, viruses have to have specific cells. Some viruses can have a generalized cell like rabies. Some of them have to have a very, very specific cell. HIV has to have a lymphocyte. Not only that, it has to have a T lymphocyte, and it can only attach on one spot, just like the coronavirus can only attach on one spot. You've probably seen the picture of the TV where it shows a sphere and has a little T thing sticking up, or sometimes a T thing with a Y on the top of it. That's to show the site where that virus can actually attach. When it attaches, it's brought into the cell, and then it destroys the cell. Okay, getting back to where I was at. Hepatitis is a virus that attacks only hepatocytes in the liver. When it first got into the human population, there was one kind, hepatitis. As it mutated, because you know we start scrambling our DNA, when it mutated so it was different than the initial one. They realized that it was going to be in the human population and slowly changing. So we started naming them according to the alphabet: hepatitis A, B, C, D. I think we're up to E now. Okay? That's what hepatitis is. Hepatitis is a virus that attacks liver cells. Okay? Hemorrhoids. We talked about varicose veins. Do you remember? And a varicose vein in the anal area or rectal area is called a hemorrhoid. Varicose veins. Gallstones. You've heard of bladder stones, kidney stones, gallstones. Okay, and what they are is we have minerals, proteins in suspension. Whether it's in our blood, whenever there's a liquid, you have solids that are in suspension. Okay? When the fluid that suspends it, which we call solvent, when it suspends it and it's too thick, it'll start to precipitate out. You've seen that within chemistry. When it does, it will attach, crystallize to other precipitate. When it does, it becomes hardened. That is a stone. So really what a stone is crystallization depending on where it's at. If it's in the bladder, it's crystallization of whatever it might be. When it's in the liver, the kidney, it's crystallization of that. And when it's in the gallbladder, the gallbladder, a gallstone is the crystallization. I'm going to put up there what is actually crystallizing, but one, lack of water, high protein, high minerals, pH off, those all lead towards a stone, a crystallization. Now, what we're going to do is we're going to shut this down. I'm going to erase the board, and I'm going to come back to the third segment, which is only the second segment of this. Good. Okay, the last segment in the digestive system is talking about digestive enzymes. Now, anatomy is knowing the names of the parts of the human body or parts of a body. And I changed, as you remember at the very beginning of the semester, I said there are some things I'm going to explain using physiology so you can see the whole picture. One of the things that I'm going to do is when we do the kidney, I'm going to tell you how it works, which is the physiology of the kidney, and you're going to be responsible for it. Another one is the digestive system. When we talk about chemical digestion, what is involved? Well, it's going to be cells that produce certain enzymes. Enzymes are proteins. So what I did for you here, and I'm going to ask you about 5 to 7 questions on the exam, is I put the stomach, the pancreas, the small intestine, intestinal juice, what is it? And I'll talk about what is stomach juice. Okay? Now, you're going to be required to know these things. So I'm going to run down to you what I have up here, and then it's up to you to memorize it. In the stomach, we call now remember the stomach is made up of simple columnar cells, but that isn't all that's in there. There's digestive cells, cells that are specialized to produce digestive enzymes. Those collectively are called gastric glands. Gastric glands. And there are 3. 2 of them are exocrine, 1 is endocrine. Remember what an endocrine was? A ductless gland that produces a hormone. Parietal and chief cells, also called zymogenic, produce the exocrine enzymes. And this is called gastric juice. So the parietal and chief together produce gastric juice. What happens is the parietal produces HCl, hydrochloric acid. The chief or zymogenic produces pepsinogen. Pepsinogen is a sleeping enzyme. In other words, an inactive form of an enzyme. You got to get rid of the ogen to make it active. HCl gets rid of the ogen and produces pepsin. Pepsin then break peptide bonds of amino acids, of proteins. It also produces gastric lipase. Gastric lipase breaks down a little bit of lipids. Remember, most of it comes from the emulsification to bile, and you're going to see the pancreas takes care of it. But a little bit by the stomach, not much. Gastric lipase, ASE means it's an enzyme. What controls this is when the stomach stretches, it produces a hormone called gastrin. Gastrin is produced by specialized cells called G cells. G is in the letter G cells, which collectively are called enteroendocrine. Enteroendocrine cells, G cells, produce gastrin. When in the presence of gastrin, parietal cells secrete HCl, chief or zymogenic cells produce pepsinogen. In the presence of HCl, pepsinogen becomes active in a form called pepsin, and that breaks down the proteins. Next one, pancreas. Remember we said pancreas was exocrine and endocrine in function. The ones you see in black right here break down proteins. Remember us talking about that. One of the most difficult compounds to break down is a protein, and it's breaking that stupid peptide bond. We do it with an acidic environment. We do it with chief and parietal cells. And then in the pancreas, we hit it again with these major four. They break down proteins. Pancreatic lipase breaks down lipids along with gastric lipase. And then pancreatic amylase, again an enzyme, breaks down carbs. So this is carbohydrates. This is fats. This is proteins. Nucleic acids are coming later. In the small intestine. Now, remember us talking about the small intestine. I said we think that the small intestine is for absorption of the pearls, absorption of the nutrients. And I said not with the duodenum. The first part, which is about 10 inches or so, the first part of the small intestine to the duodenum is for chemical breakdown of everything down to the pearls. If it gets out of the duodenum, it's going to come out as feces because it cannot be absorbed. And I put two things here just to recall, wanted you to recall that we haven't lost sight of what we learned when we did the micro portion of anatomy. Remember the goblet cells that produce mucus? And then the simple columnar cells and villi and all that stuff, that's still true because we're in the small intestine. And then we have intestinal cells or glands. So in amongst the simple columnar cells and the goblet cells are what we collectively call intestinal cells or glands. One of those types is Paneth. Paneth cells. That's a type of an intestinal cell, and it produces lysozyme, and it's phagocytic. Now, you remember what phagocytic was. You eat, cell eating. Lysozyme, remember lysosome inside of a cell for intracellular digestion? Lysozyme is an enzyme that breaks cells down, usually bacteria. You also see enteroendocrine again. Okay, we know that endocrine produces hormone, and in the stomach it was gastrin, G cells. Here it's S and K cells, and it produces secretin. Secretin. So when food is in the duodenum, it stimulates the enteroendocrine cells to produce secretin. In the presence of secretin, it starts releasing all of these digestive enzymes. Now, I forgot and erased it, but I put it up here again. Cholecystokinin, CCK. And I was going to tell you about it when we were talking about bile. we have usually with all foods, we have a small amount of lipid intake. And that is going to be emulsified by bile. Remember the bile coming out of the hepatic ducts and it went into the cystic duct for storage? Well, it could also go straight down the common bile duct. Let me show you. This is the gallbladder. This is the cystic duct. And then it's kind of a space here. And you can see a little green duct right here next to the pancreatic duct. That is called the common bile duct. It takes bile that's produced by the liver and it drops it into the same place that the pancreatic duct does. That's for emulsifications of fat. Now, getting back to where I was going with this. You're going by KFC and you can smell that delicious chicken. You've just got to have some. So you go in and get yourself a bucket and you start gnawing on the chicken. Now you know when you look down the bottom of the bucket, it's floating in fat. That fat is now inside of you, which means that constant little drip that's coming out of the hepatic ducts is not going to be enough. So you have to go up to the the gallbladder and say, hey, I need some help. I need to emulsify all this fat this dude is now taking in. So the the gallbladder has to squeeze like a sponge to get all that bile to go down the common bile duct to enter the duodenum so it can break down the fat. In order to do that, it's under the influence of a hormone. Cholecystokinin, CCK, is a hormone. So what happens is when fat enters from the pyloric sphincter into the duodenum, it triggers the release of CCK, and right around the intestine is capillary beds that loop it right back up, called the hepatic portal system, loop it right back up to the liver. Once it gets to the liver, attached to the liver is the gallbladder. It constricts, shoots the bile down there, and emulsifies the fat. So CCK is a hormone that stimulates the gallbladder to constrict, forming a high concentration or releasing a high concentration into the duodenum itself. So it's a quick little loop. So now you know what CCK does. Also, remember we said one of the other problems we have is when the pyloric sphincter opens up, that it wasn't bolus anymore. It was called chyme. It's very acidic, pH 1 to 2. So we have to neutralize it. And we said that sodium bicarbonate is released, and also the Brenner's glands, which are collectively called duodenal glands because that's where they're at, but they're called Brenner's glands, release sodium bicarbonate also, alkaline. Okay, now do you have these? You have to know what the cells are in the stomach and what they do. The pancreas, what they break down. Okay, proteins, lipids, carbohydrates. Small intestine, this was just so you would remember what the small intestine is made out of. Intestinal glands, PNF is one type. Lysozyme is an enzyme that breaks down cells. Phagocytic, eats the cells. Neuroendocrine is a hormone. S and K are the names we put to the cells what we name the cells that produce secretin. Cholecystokinin is another hormone that is abbreviated CCK that is picked up by the hepatic portal system and causes the gallbladder to constrict, releasing bile. Duodenal glands, the specific ones I brought up here is Brenner's to neutralize that pH. Now back to the intestinal juice now. Intestinal juice. Lining the intestine in the duodenum are cells that produce enzymes collectively called brush border enzymes. Okay, those are enzymes that are going to break down carbohydrates, proteins, and nucleotides. Once you get here, you should be a monomer. If you're not a monomer, you're going to become feces. Okay, and one last step. Again, physiology. We were talking about the liver breaking down red blood cells. And we said red blood cells, remember you got the phospholipid bilayer, yes, phospholipid. We also have hemoglobin, remember, which is an enzyme that oxygen attaches to. So when you break down hemoglobin, the globin, which is the protein part of it, can be recycled or excreted. And the heme portion becomes a compound called biliverdin. Biliverdin. Now, this is going to go down the gallbladder, the common bile duct, to the duodenum. In the duodenum, there's a bacteria. No, take that back. In the duodenum, there is an enzyme that converts biliverdin into bilirubin. And we hear of bilirubin because a buildup of bilirubin in the blood is a condition called jaundice. Now there's bacteria, now you're in the intestine. There's bacteria in the intestine that causes bilirubin to become urobilinogen. Urobilinogen. Bacteria have made bilirubin into urobilinogen. Urobilinogen has two fates. One, it can be picked up and taken to the kidneys and excreted as urine as urobilin, which gives it the yellow color. Urobilin, the yellow color. Or it can stay in the digestive tract. In other words, it isn't picked up by the hepatic portal system, and it can come out in feces as stercobilin, which gives it the brown color. Okay, we're good.